So welcome to another session of the Size and Growth Zoom series that we're having here. Again, we have two good talks today. The first is Jonas Cremer, who is a new faculty here at Stanford, and the second we'll get to is Naama Barkai from the Weizmann Institute. So why don't we get started with Jonas's talk? Want to share your screen? And
That's good. Can you see this? Great. Good morning, good afternoon, good evening, everyone. Thanks for logging in. Since this is a series about cell size and growth, I thought, let's talk about our recent work together with the lab, where we thought a bit about the relation of bacterial swimming and cell size. But since many people are maybe not so familiar with what bacteria are doing in cell size, let's start really with the basics. If you grow, for example, E. coli in different conditions, they can grow with different rates, different speeds, or different growth rates. And if you then look how the size of these cells are changing, you see that faster growing cells are really bigger than slower growing cells, at least in steady state growth condition. And that's really a famous observation, first observed in Salmonella. And I would say that's still one of the most fundamental relations which brought people to think about cell size control in bacteria and how is really this relation working between cell size and growth, right? And there are many great talks also in the series to really try to understand cell size control and growth, of course. But so what I want to talk about today is how is this relation important when we think about swimming and cell growth. And as you will see, we will come back to this fundamental relation at the end, and without taking this relation into account, basically we cannot really understand motility gene expression. And as I said before, so this work really started or was done mostly in the lab of Terry War, and it was a great collaboration with him and also with Tomo Honda, who is now at the Berkeley National Laboratory. So to get started, because also many of you are probably not so familiar with bacterial swimming, let's really start with the basics here as well. So many bacteria can swim, right? So they rotate flagella, for example, and then they do this run and tumbling movement to swim. And that's only one thing they can do. They can also sense pretty well the environment. So E. coli has, for example, several receptors to sense amino acids and oxygen. And then they can, of course, integrate this. And this is really where the magic is happening, because they can do chemotaxis, right? So they can sense gradients and then coordinate their movement to swim along the gradients. And this has been worked out really in a lot of details for E. coli at least. And it's really great to see how much we understand already on the molecular level about this process. But then we started this project with this question: what is really the physiological and ecological role of swimming? So can we say a bit more how the swimming apparatus is integrated to whatever else the cell is doing? And can we even try to start to speculate why swimming is so important besides the obvious? I mean, fine, it's good to move, right? But can we say a bit more, right? And the important thing here to realize is that bacteria actually shape the environment, right? So to understand that, consider really two simplified scenarios. One is the bacteria is just sensing the environment it is encountering, for example, some chemical gradient given by whatever other process, right? But then the other extreme is actually that other processes are not really shaping or setting any gradients, but you just have an attractant or something the cell can consume, cells are consuming them, And when cells are consuming them, they form the gradient and then they move along the gradient. And it turns out that the second scenario is really so important, and bacteria really readily run into this scenario. And basically there are two reasons. The first one is the sensitivity range of these receptors cells have is very narrow. So then cells easily shape or go from one extreme to the other of this range they can sense. And then this gets combined with a really high uptake rate for many of these chemoattractants. E. coli can sense that well. And so this is really like a very common scenario, and therefore you can readily see this in the lab. If you just start with a droplet of cells in the middle of an agar plate, and you see that cells are moving out. And people have studied this since a long time, but how the process really works was kind of unclear. And so what we did is really a quantitative study to understand how this collective movement of cells is working and how cell growth is so important in that process. So what you see here is a ring of cells going out. So they swim out here. Over time, you see the ring getting bigger. And we really did a quantitative study observing how cells move along the self-generated gradient at the front of the ring. And we tracked this by microscopy. And then I would say the biggest contribution is then integrating this with a model to really entangle the process, how this works, right? And I don't have time to go into details here, but it's really this combination of movement, the shaping of the gradient, and cell growth, which is so important. And then overall, what we were able to do is to quantify what is kind of the fitness advantage of a population when they grow like this and when they expand into new territories. And the important thing here to realize that this is a really powerful expansion process and really important for cell growth if cells swim in nutrient replete conditions, right? But then actually what we wondered then is, well, how relates this to the regulation of the motility machinery? Because while we saw that cells swim in nutrient conditions, the common picture out there is still, or was at least, that actually swimming is more a starvation response, at least for E. coli in the studied strains. And this picture was so strongly supported by gene regulation measurements. And so to understand that, so the E. coli flagellar apparatus has a lot of different proteins, right? It's distinguished into different classes, 1, 2, 3, but really class 2 is, for example, all the structural elements. And so to understand if motility genes are expressed a lot or not, or if the flagella is expressed or not, we can look at how are these genes expressed. And I show you here an example for FlyA, which is a sigma factor to express motility genes. But really, if we do mass spec and analyze other genes, or take mass spec data and analyze other genes, we see this confirmed. And so what you see here is, if you take a reporter for FlyA, you see that the expression of motility genes is really lower at fast growth rate and much higher at slow growth rate, right? So that really suggests that, well, maybe swimming is a response happening more in nutrient poor conditions, right? But what we want to say here, or what I want to convince you, is that it's not so simple, and we have to think actually about what does this unit here mean, right? But the first thing we can do to understand this better is to actually look how cells are behaving in different growth conditions. And for that, we really advanced common tracking assays to really get also the fraction of cells out. So what we did is we grew cells in batch culture, we took microscopy, we got these tracking patterns, how cells are moving over time, and then we analyzed for each single cell a swimming speed. And so what we get with this assay is a distribution of swimming speeds what's the average swimming speed, and how many cells are swimming. And so we can do this for different growth conditions. And if we do this for different growth conditions, what you see here, for example, growth in glucose, which is fast, growth in glycerol or mannose, which is not that fast, you see basically the same pattern, meaning cells are all swimming at pretty fast growth, at pretty fast swimming speed, right? And so if we check this for even more growth conditions, we really see that our cells are swimming. All of them actually are swimming. The fraction of motor cells is really, really high, and the swimming speed is not changing that much, right? But then how is this related to actually to this gene expression pattern and how motility genes are expressed, right? So why is the expression of motility genes higher at slow growth compared to fast growth, right? And the resolution is really simple, and since I'm talking here to experts thinking a lot about cell size regulation, let's put the answer right out, right? So it all has to do with thinking about cell size and what this unit here is, right? And to see this, let's look at this relation again more quantitatively. So you see here an exponential decay, and what we are looking here is at a LAC reporter of gene expression. And so what that means is it's a readout for what the relative expression of this gene, or maybe to easier explain this, if you would take mass spec data, what you would plot here, what this axis would be, is the fraction of motility of proteins being motility genes, right? And so that really is going down with growth rate, right? But then we have to consider the fact that cells are changing their size with growth rate, right? And so that's really where this relation I showed you at the beginning comes in. So cell size is really changing a lot, and cells are getting bigger and bigger with faster growth, right? And so if we consider this now, and instead of looking at the gene expression per biomass, which is plotted here, but we look at gene expression per cell level, then we actually see that these trends perfectly cancel each other, and we get a flat line, meaning the expression in our bacterial strains, the expression of motility genes is really not changing at all with growth rate, right? And that might make a lot of sense in terms of, you know, what is needed for the cell to swim and to maintain really motility. And to understand that a bit better, we can now use a construct and change motility expression levels, right? So we can dial up and down motility expression and see how the cell responds, how the swimming is changing. And so that's what we did. So FLU-DC, FLHDC is a master regulator of the swimming genes. And so if we use a titration construct, and if we titrate this gene, then as a consequence, other motility genes are also changing, and we can then observe what is happening with motility. So what you see here is that our reporter we used as a readout for motility genes is really showing that if we titrate this FLU-DC, then motility genes are less expressed. So for example, let's take here different colors mean different growth conditions. Let's take one growth condition here, where if we start here with the Y type and then start to, if we start, it's the other way around, right? So if we start with no inducer level, right, then we are here. But then if we increase the inducer levels more and more, then we reach the Y type levels with a lot of motility genes, right? And so now we can basically take this relation we see here, and we can just say what would happen in the scenario where the expression of motility genes would not change with growth rate, right? So we can just use different growth conditions and pick different inducer concentrations. And if we do this, if we take here, for example, one level, then we see that the motile fraction of cells is really going down at slower growth rate, right? So this is simply making the point that this upregulation of motility genes is just needed for upregulation at slow growth is just needed so that cells, stay motile at slow growth conditions. And we know already the reason is compensation for cell size. So that brings me already to the last point I want to make, last major data point I want to make, and that's we also counted then the flagella number directly, right? So we saw this result that gene expression is not really changing over growth rates when we look at gene expression per cell. So we can therefore also directly count flagella number. So what we did is we stained flagella for different cells growing in different conditions. So here you see two examples: growth in mannose, which is slow, cells are small; growth in glycerol, cells are large. And we count flagella number. So we can stain flagella and we can count them. And then we can look at the average flagella number for different growth conditions and how the average flagella number relates to motility. And this plot is a bit complicated, but I guide you through. So this is the average flagella number, right? And the important point here is that all these circle points are wild-type strains. So you see for wild-type strains, for the wild-type strain in different growth conditions, you see that the fraction is high. So cells are really all motile, even though gene expression per biomass is really changing a lot, right? Cells are all motile, and the number of flagella is roughly between 4 and 5. But then if we use our titration strains and we don't do this upregulation at slower growth, then we see that the motile fraction really goes down. If motility expression levels are too low, the motile fraction is just going down. And that's kind of almost trivial now, given this understanding we have with the cell size control. But the important point here to see in this plot is that actually the regulation of flagella genes is working kind of in an optimal range, right? So the wild-type flagella is really expressing 4 to 5 flagella per cell, which is really optimal in a sense that all the cells are motile here already with 4 or 5 flagella, but the cell is also not expressing much more flagella than needed, right? And so Mike, I realize I don't have a clock, so you have to remind me how much time I still have. Or 20 minutes, so perfect timing. Okay. So this is really the major result of this study, right? So our strain, we studied our strain, it's motile across different growth conditions, but really the regulation appears to be optimal. This number of flagella is really maintained across different growth rates and cell sizes, so that all cells are motile, but not more flagella than needed are expressed. And this is, again, the summary. I will do this quickly so that we have maybe 2 more minutes to discuss what this means. Again, cells swim in nutrient replete conditions. If there are nutrients around, the cells swim, especially or also when growth rates are really fast, right? But then to accomplish this in an efficient way, cells adjust the expression of their motility genes with cell size so that actually the expression per cell remains the same. So when I talk about adjustment, it's adjustment per biomass, right? But the expression per cell size is actually the same. And then finally, this is really efficient because the number of flagella appears to be optimal to maintain a full motile population. And so this kind of brings me to this point, which I wasn't really aware of before going into this direction, because I haven't really thought too much about cell size. But I think this is a neat example showing how really, if we think about the cell and how the cell works, then really nothing makes much sense if we don't think about size. And these interpretations out there with motility being a starvation response, that's one example, right? It's I think highly misleading. All right, and so finally, given these results, I think they are now almost like so clear, and I think, yeah, I think resolving this puzzle, but then they are also really interesting as a starting point to think more about the relation between cell size and growth in general, right? Because so we have this famous fundamental relation here, but we also have then, as I showed you, motility genes being highly expressed on different levels per biomass at different growth rates, right? And so smaller cells express more motility genes than larger cells, and that really comes with a huge cost, right? So if we knock out here the motility apparatus and we look at different growth rates, then we see really growth rate improvement by 20% in the slow growth condition compared to 10% in a faster growth condition in glucose, right? So if you compare here, a point here and here, right? And so what that means is that this size-dependent regulation of flagella genes, right, really couples immediately into growth, right? And so if we want to think about the relation here between cell growth and cell volume and growth rate, right, then we have all these effects here at play that cells adjust the expression of many genes size, and that really couples into how growth depends on right? And so that's one thing maybe we can think more about now that we have this understanding. And but then the other thing, of course, the maybe even bigger question is, how is the cell being able to maintain flagella numbers so perfectly across growth conditions? And that's really where the regulation magic must be happening, right? Okay, with that, thanks for your attention, and happy to take questions.
Thank you, Jonas. I think most of us would agree with your conclusion about cell size and it being
good.
Yeah. So we have a bunch of questions in the chat. If you have a question, you can post it in the chat. I will call you out and you can unmute and ask your question. And if we don't get to it, which will stop close to the half-hour mark, everything will be posted on Slack. So Jonas will have a chance to respond to you there. So let's just start going through them. So the first question we have is from, sorry if I pronounce this wrong, Srividya. Would you like to unmute and ask your question?
Hi, I had a couple of questions. Should I just go ahead and ask them back to back?
Go ahead, yeah.
Okay, thanks for this interesting talk. My first question was, how does the growth context, for example, agar plate versus liquid media, matter for the specifics of the swimming growth trade-offs? That's number one, growth context. And number two, how does cell shape affect these results? Have you all done experiments with anything other than rod-shaped cells, or more specifically E. coli? Thanks.
Okay, so I mean, what I can say is for E. coli, and that's maybe I start with answering the second question. So we only looked at E. coli up to now, and already for E. coli, I would say we have to understand much more, because we're looking at certain E. coli strains here, and already there's interesting to understand actually what other E. coli cells are doing. Given these high costs here, right? Yeah, the short answer is really we have to measure more in other strains, but even in other E. coli strains, right? But I can answer much better the first question: how is swimming behavior changing in liquid and in agar conditions, for example? So at least for our E. coli strain, so what we've done is we've done these experiments in agar and in liquid culture, and we use really microscopy to track in agar, but then also in liquid culture. And the short answer is we really don't see much of a difference. I mean, of course, if you look at diffusion rates, these diffusion rates are used to effectively describe tumbling behavior. That's changing in agar concentrations in agar, because cells bump into, the agar mesh all the time, but the swimming speeds are not changing much.
Okay, thank you, Jonas. Navish, would you like to ask your question?
Sure. Hi, Jonas. Really nice talk. I had a question. So thinking about the relationship between cell size and motility, so there's also another factor which comes to mind, which is the hydrodynamics, right?
Yes.
So I was wondering, like with the smaller cells, they still have the same number of flagella and they swim at the same speed, but since they are smaller, the drag must be smaller. So shouldn't they swim faster?
So yeah, so that's a really good question. So we don't have a good answer to that. I mean, basically, I think if you look at the flagella and the cell, right, the flagella are much larger than the cell itself. So I don't have a good intuition why the cell size is not affecting swimming speeds that much, but that's I mean, these are the observations we have, right? And so, yeah, but it's really important that we think more, I think, about the relation between, friction and hydrodynamics and how that relates to cell size.
Okay, thanks.
Hey, Jan, would you like to?
Yeah, I mean, wouldn't the hydrodynamic drag just be determined by the length of the whole thing plus the flagella? So maybe like the length of E. coli plus flagella doesn't change that much. Anyway, just a thought.
Yeah,
That wasn't
that's roughly true.
a comment from the previous question. My question was, it's really striking result that there's this sort of constant amount of flagella gene expression regardless of growth condition and cell size.
Right.
And so I was wondering if there was, feedback on the expression of that protein. So like in your, I don't know, maybe you could see that in your induction system. Like if you look at the expression from the, endogenous protein when you, or, by changing the levels, can you see any evidence of any kind of feedback regulation that's maintaining this constant amount of flagella gene expression? And sort of a related question to that is, here, like it doesn't seem like you ever got more than the canonical number of flagella. So I was wondering if it was possible to overexpress those genes.
Yeah.
And or if you did overexpress them, but there's some other kind of like steric hindrance constraint that prevents more flagella.
I mean, these are both excellent points, right? So so to the first point, when we use our inducer system, we really break this symmetry, right? So and our inducer system is really working titrating the master regulator FLHDC, right? So all the magic in terms of regulation and how this is coordinated so nicely with cell site should therefore happen on the regulation level, regulating how much this master regulator is expressed, right? And I think it would be really cool to go more into that direction to really understand how the cell is doing this so well, right? And maybe this will provide deeper insights into cell size regulation in general for E. coli, right? And then to your other point, if we weren't able really to express more than these 4 to 5 flagella, right? And I mean, we also haven't really tried a lot, but we just observed it at least in our chromosome-based expression systems. We weren't able to go over that limit.
But you think that's not due to some negative feedback or anything? I mean, I guess how would you even have the negative feedback on your synthetic system unless there's some
negative feedback preventing the expression to be higher? That could happen, right? But we don't know at this stage. Yeah, we don't know.
Thanks. Great talk, by the way.
So how about we take just one more question and then we can move the rest to the Slack channel. Martin, would you like to, or I'm sorry, I skipped. Marco, would you like to ask your question?
Hi, can you hear me?
Yes.
Okay, so great talk. I was wondering what says the fraction of motile cells, because if I think about it, it could be many things, and I cannot get a clear idea of where it
Oh
could
yeah,
be.
I was very short in I basically skipped the definition. So you're meaning what do we call the fraction of motile cells here?
No, not, I mean, I understand what we call the fraction of motile cells, but I don't understand. I mean, there are many factors that could set this fraction, physical and biological. What really determines this fraction in a population? So that
Right.
I can understand the liquid size, right?
Well, so yeah, I mean, we just observed these cells in liquid culture and also in agar and just looked at how motile they are, right? So that's just an observation at this point. But if you are
Okay, so
asked,
but you have like an hypothesis of what could make them, what would set this fraction of motile cells, which then would give me more insight in what's going on here.
yeah, so that's basically what we try to do with our expression system. So we think that a huge factor determining the fraction is really the expression of the motility apparatus. So if the number of flagella just falls, then at some point, you know, some flagella have some cells have flagellas, others don't have flagellas, and that's basically what we see in our expression system. Where, so these are wild type levels here, but then if we take here wild type point and change expression levels, go to lower expression levels than wild type, then we see that the motile fraction is changing. And I don't have a plot here with flagella number, but we counted that as well. So you see that the number of flagella is really going down. And at some point you end up with more and more cells having
So
no
it's something
flagella.
to do also with the variability, cell-to-cell variability in gene expression. Right?
Yeah. So good point. So effectively, then we see, if you wish, phenotypic heterogeneity, if you want to use this word. We don't know really how it comes about. I think if it could be a lot of variation of gene expression from one cell to the other, but it also could be a fairly constant gene expression, and then you know, the number of flagella is just unevenly split during cell division, and that can already lead to changes in motility fraction. And
Okay,
we haven't investigated this further at this point.
thanks.
Okay, thank you for all the questions. I'm sorry we didn't get a chance to get to all of them, but we should probably move on to the second talk. We're almost five past half hour. So Naama, if you're there, could you share your screen?
Okay.
Yep,
Okay.
looks good.
Great. Thanks.
Mhm.
Can I start?
Yeah, go ahead.
Okay. So I'm going to discuss actually published work. It's work of mostly Eyal, Moshe, and Gilad. Eyal and Moshe are former graduate students. Eyal is now a postdoc in Stanford and Moshe in Princeton. And Gilad is still a close colleague. And I will also touch just a little bit about work of Felix Jonas, who is a postdoc in the group currently. So we're thinking about the growth rate and size, and as everyone is well appreciated, those are sort of, if you want, global features of the cell physiology, and they are result from all this complex interplay within everything that is happening inside this. But we're going to take a bird's eye view, like I think many people probably here are doing. And we're going to look at very simple models of the cell. And the way I structured this work is basically I'm going to talk a little bit about, I think, things which people might know, And this is about a very simple model of cell growth and how it actually determines the limits of cell growth, how fast can cell grow and what limits it at the best optimized condition. From there, I will go on and show you some data that we have in budding yeast about these particular questions, and I will investigate it. And from there, I will go into sort of maybe suggest a similar limitation of cell size, which is less strong than the limitation of cell growth, but is actually discussing some interplay between the two. And the way we're going to think about it is just as a very simple machinery of making protein. We have ribosomes, we have protein, cell size is proportional to how many proteins we have. And the growth rate is basically depends on how fast we're going to make those proteins. And of course, the simplest thing is we still have to have this autofeedback loop. Ribosome not only make the protein, but make also themselves. So this is a very well known model, very simple, clearly ignores everything inside the cell, but still it has some predictions which are interesting. And the prediction is that if we just take the only two parameters in this model, which is the translation rate and the ribosome fraction, meaning what is the ratio of ribosomal protein out of the whole proteome, roughly speaking, we get that cell growth, or the specific cell growth, which is a rate, the specific rate by which a volume of the number of protein is increasing, is just proportional to those two parameters, to the translation rate and to this ribosomal fraction. Actually, if you do this normalization correctly, it equals. And one clear and immediate consequence of it, which is well realized, is that cells can never grow faster than this translation rate. Of course, it cannot even get to this translation rate. This would have been happening if all the protein in the cell would just have been ribosomal protein, which of course doesn't make any sense, doesn't happen this way. But this is sort of the upper limit, cannot go any faster. And the larger the ribosomes out of the full proteome, the faster the cell would grow. So this is sort of the fundamental limits, which I think is very well realized. And this is what we're thinking about it. This is sort of the mechanistic limit, how fast the ribosome actually grows. One ribosome goes over one mRNA to produce the protein. This is sort of the mechanistic issue. And this is, in a way, sort of determines how fast the cell as a whole can grow. And what about size? Size doesn't go into here. Basically, there's no statement about size. Size, to actually include size here, we have to think about division. And we don't say anything about when cells divide. We can think about all of them just growing proteins forever, making cells as large as possible. There is no, in this specific formula, there is no limitation about size. Now, does this simple model makes any sense? And we know from many works, including, I think, the very beautiful and fundamental studies of Terry Waugh, but actually, if you look inside cells, you see that this basic prediction of this model actually holds. This is actually our experiment. We did it in budding yeast. And what you see here at each point is cells growing in different conditions. So we change conditions, we measure growth rate, and we measure the proteome. Actually, our data is only the solid data. The open data points are taken from another group from Harvard that published the proteome data and growth rate. And what you see, it's very nicely, independently of where the work is actually being done. There is a linear relationship between the proteome fraction, namely the ratio of the ribosomes. No, sorry, I'm home, it's late here. The proteome fraction, namely the ribosomes, the fraction of ribosome out of the total proteome as a function of growth rate, just as was predicted, and not only that it follows a linear prediction, but also the slope is as predicted, namely the translation rate. There is one particular point to notice is that this curve, at least in budding yeast, doesn't go to zero, but actually goes to a finite point, which we interpret as having some reservoir of ribosomes, if you want, namely ribosomes which are not being used, under this growth condition, and we can discuss this. We can see some conditions where this can be used. But I think in principle, one can see that this very simple model, not only in bacteria, but also in budding yeast works pretty well. Okay, so this is about cell growth. And the question is, can we go further? Is there also fundamental unit, mechanistic unit, just like this translation of translation rate, the working of ribosome of mRNA, that also talks to cell size. And the question is how to approach this. And the way that we thought about it is the following. So, you know, translation rate is one parameter that is needed to making protein, but there are other biosynthesis processes that are required, like making mRNA, like maybe making amino acids, and each of them can also have some fundamental unit, mechanistic unit that can then talks to this higher order physiology, for example, cell growth or cell size. And the idea is that if we have such a unit that is limited by protein production, that limits protein production, then it will probably be evolutionarily optimized. And when we say evolutionarily optimized, it will probably be working at maximal capacity. Now, what does it mean, maximal capacity? It means that if we now add some demand, so we grow the cells in some environments that were, you know, adapted to, and now we increase the demand for protein expression under this particular condition, then this particular process, which is evolutionarily optimized and working at maximal capacity, will not be able to adapt. And we can then maybe detect it. So if we ask what processes fail to adapt when forcing increased protein production, maybe that will give us some more insights about what actually is the limiting factor. For example, if translation is the only thing which is limiting, and we force the cells to make much more protein, then we expect to see a signature that is sort of reminiscent of of inability to increase this translation capacity. So how do we do this? So this is the idea. We actually, that's how we started. The idea was, and also again, it's not our idea. People have been doing it for many years in different ways, but we did it sort of in a systematic way, simply force the cells to make lots of protein, and we did it through making a library, which is called library burden. And this is by taking M-cherry protein and sort of something that the cells doesn't need, but doesn't affect the cells, drive it by the highest, if you want, highest promoter in the yeast cells, TDH3, and use an integration procedure that puts it into the genome in multiple copies, which is actually variant. So by one transformation, we can get a whole library of cells that go from 1 copy to 20 copies. This is sort of what the cells look like. One of them, we simply go pick up the colonies that show that being red, look at them, the cells look kosher, no particular problem. And we can get a library. Here is, for example, each point here is basically one colony that we picked, which has a different fluorescence. This is when we just have 1 copy. This is, I think, when we have 20 or 25 copies, I don't remember. And what I show you here is that basically the growth rate or the division rate as a function of the fluorescence. And you see that it goes down linearly, which is sort of what's expected. You force the cells to produce more and more protein, growth rate goes down. Now, just something to note, we believe that 1 copy is about 2% of the proteome. So it's really, we go very high and still we get about 15% reduction in growth rate. And we sort of interrogate this library in different ways. And we show that we get a combination of both translation and transcription processes that affect this reduction in growth rate. But this is not the story that I want to focus on today. So we have this library, and now we want to know what fails to adapt. So what fails to adapt? If we just look again at the proteome function of growth rate, we see that it goes, each line here is just different condition, but the lines in the same color are the burden. You see that basically they keep the same. If we focus on one burden, they keep the same slope, which means that the translation rate doesn't change, but it goes down, which means they make maybe more use of these available ribosomes, but basically translation rates in the specific translation rate is the same. Okay, but like I said, what we're really interested in is looking for additional limits. And the idea is that those additional processes, biosynthesis processes that are limiting will fail to adapt, and we'll be able to detect them. So how do we detect them? So the idea was when something fails to adapt, probably it will have an expression signature. For example, if we basically deplete the amino acids, so the cell feels amino acid starvation, we look at gene expression, we see the known signature of limited amino acids. If we are limited in energy, then we look at expression signature, we see a limitation of amino acids, of energy or glucose. If what is limiting is only translation, then perhaps what we're going to get is a signature which is similar to signature of deleting ribosomes. So this was the idea, and we said gene expression is really a very sensitive probe of what is limiting in the cell. So what we can do is we can take a library and simply profile them, profile the gene expression, and we did this in 3 different conditions, and this is what it looks like. It doesn't matter, just to show you that we see something, for example, in SC, which is a rich media for binding yeast, top 1,000 genes which are changing. You see that as we change the burden, you see that those genes go up, those genes go down, and the same for the other conditions. Okay, so now we want to identify what is the burden signature. The problem is that we get a mix here because the cells go slower. So maybe what we see, those genes are simply genes which are growing, which are basically a signature of slow-growing cells. So the way we sort of approach this was to take additional data sets that are available in the literature, for example, environmental perturbations, which people reported both the change in expression and the change in growth rate, and genetic perturbation data set from Frank Holstich, where he mapped the profile of about, I think, 2,000 deletion mutants in binding yeast, and he reports both the division time and the gene expression. And from this, we can deduce the growth rate signature. Because, for example, if we take a gene, ribosomal protein, we see that it goes down. This is coding for mRNA coding for ribosomal protein. As we increase growth rate, as we decrease growth rate, division time goes up. This gene goes down, right? On the other hand, SIS1, which is a stress gene, when division time goes up, this is increasing. And we see the same behavior also in the genetic perturbation. What happens in our data set? In our data set, we see that the ribosome doesn't change as a function of growth rate or the copy number in the burden cell, in the burden library, but the SIS1 actually goes up. So now we can ask if the signature that we see is the same like the signature of growth rate. The answer is no. If we compare the genes, the behavior of genes in genetic perturbation to that environmental perturbation with growth rate, the ones that are going up or down, we see a very clear correlation. So this is a growth rate signature. When we compare the signature to our burden cells, we see that it's mostly unrelated. We have some which may be explained by genetic, by this growth rate signature, but the most of the things that we see here are actually unrelated. So we have something unique here. So we tried to look at this unique thing, and we looked at the ribosome genes. Nothing changed. We looked at lots of nutrients limitation. We didn't see anything. We did see HSF1 target, which has to do with protein folding. So they go up, which actually makes lots of sense. But this didn't sort of lead us too much. So what we decided to do was to compare it systematically to what happens in mutants, right? So we can take mutants, and we ask if when we delete this mutant, do we see the same signature like we see in the burden strain? For example, like I said, if what we do is we limit ribosome, then it would make sense that if the ribosomes can adapt, then it would make sense that a strain deleted of ribosome will be similar to our burden signature. If what the burden is doing is depleting amino acids, then it would make sense that the signature that we see is similar to the signature that we see when we delete GCN4, which is a regulator of amino acid biosynthesis. So like I said, we took the data of Frank Hostage. He has about 2,000 deletion mutants. So each of the points here is one deletion mutant. And what we do is we compare the signature of the deletion mutant with the burden signature. And what I highlight here, so we have lots of mutants, they behave differently. We have some which seem significant. We don't yet know what they are. We'll wanna know soon. But the first thing that we looked at was this, all the proteins that have to do with ribosomal RNA processing, which have to do with translation. And as you see, they basically fall within the cloud. There's nothing special about them. So they don't seem to be sort of the dominant thing which are limited. What did come up were actually mutants in the transcription machinery. And I show you here two classes of mutants. And they seem to work with a translation initiation. There's a Sargas with SNP, and what seems to be most dominating was the actually the mediator, which has to do with the translation initiation. So it is true. Is there any other indication that the burden talks to mediator deletion? So we decided to look if there is a genetic interaction, and I'll just take you very quickly through our assay. The mediator has multiple subunits. If we delete the kinase unit, we get flocculation, and this can be compensated by other deletion in other subcomplexes of the mediator. We can see this very nicely when we measure flocculation. And the main point to see is that we can basically imitate the function of the head and tail subunit by the burden. So there's clear genetic interaction between them, and we can show it in other ways. So the first guess was, okay, what the burden is doing, basically mediator is limiting. There is not enough binding of mediator to gene promoters. It's a central factor required for transcription initiation. So we measured the binding of mediator to promoter in burden and non-burden cells, and this wasn't the case. We see very change in binding, and basically maybe we get 5% depletion. Doesn't seem to be enough to explain. So what is actually happening? So to actually happening, we looked at what is the common signature, what is really this common signature between burden and mediator. And what we noticed is that basically the strongest effect of the data was that we get an asymmetry in changing in expression depending on the absolute level of mRNA expression. Basically, and this we also see in the mediator, basically what we see that we have some effect that low and mid-expressing genes, which are here, are affected less than high-expressing genes. Now, what is happening here? We don't have normalization. We don't know the absolute expression here. So we had to measure it to know. And what I show you here is basically the following. The burden cells contain more mRNA. So if you just measure the total mRNA in burden cells compared to non-burden cells, the burden cells compare more mRNA. We force protein production, we get more mRNA. And this may be explained by the fact that those cells are larger. We saw it, and Tariwa saw it also for bacteria. The cells are simply larger. So the cells are larger, they also contain more mRNA. So what does it mean? If they contain more mRNA, initiation increases. So they have more initiation, and this explains why we have more expression. However, this is not uniform. This higher initiation fails at highly expressed genes. So total level of mRNA goes up, and this is seen in the low expressing and mid expressing genes, but we don't see it so much in high expressing genes. At least this is what seems to be happening in the signature. Okay, so this got us to think about transcription rate. It looks like transcription rates are problematic. In those very highly expressing genes, they might be difficult to increase. And we know the transcription rate indeed are defined by initiation rate, at least mostly, in most of the mRNA. Well, it's not true when we get to the maximal rate. So what is a maximal initiation rate by which transcription can be initiated? This is basically depends on how fast the polymerase empty the place where it's bound. So basically a polymerase binds, needs to go, and a new one comes. So the maximal, there's a fundamental limit about production of protein is basically defined by actually the elongation rate and the size of the polymerase. And now if we look at the numbers, and also people did it previously in other species, it looks like highly expressed genes are actually expressed at maximal rates. It's difficult to be conclusive about this because there are very many unknowns about how to actually measure this. But I think looking at the number from multiple points of view, it seems that, and other people reach this conclusion also, that highly expressed genes are expressed at maximal rates. And if they're expressed at maximal rate, this might explain what we see, basically, that they cannot increase further when we basically force some increase in, for example, the initiation factor, like the mediator. Okay, so now this is the last thing that I want to start speculating, why to work at maximal mRNA rates. So I'm making here some kind of leap, logical leap. What I told you now is sort of an explanation of what we see. What we definitely see is that when we burden the cell, there is a problem for the cells to adapt its pattern of the transcription. It's similar to the mediator, and it seems to be different for highly expressed and lowly expressed genes. Now I'm making this leap of claiming that it might be explained by the fact that highly expressed genes are actually expressed at maximal mRNA production rate, and the question is why. Okay, so let me propose to you why. I'm going back to this question of what limits cell growth. And when we said that we have these fundamental limits about translation rate, we basically, and we wrote down this equation, we basically assume that mRNA is not limiting. We basically, I told you, we can go as much as we can. We can always basically make use of all these ribosomes, because mRNA is basically not limiting, but of course it doesn't have to be this way. mRNA could be limiting, and when mRNA is becoming too limited compared to how many ribosomes we have, then of course we cannot use them, and we might be getting into trouble, and maybe this is where size comes in. So this is what I want to propose to you, and maybe some point to think. So we all know that when cells are growing fast, they are larger, okay? This is sort of an evolutionary tuning. What I want to actually suggest is that for a given growth condition, actually the cells can only grow, and the cells, so if for a given growth condition, and suppose that the cells need to optimize something, they could optimize growth rate, they need to optimize something, so they need to have some optimal protein allocation. So what I want to claim is that for a given condition, given that the cells need to have some optimal protein allocation scheme that is set by evolution, then in this case, there is only a critical size that the cells can adapt before basically losing this optimality. And the idea is actually quite simple, and it's coming immediately from what I tried to claim. So suppose that we have some certain protein allocation. We have certain protein allocation. We have sort of mRNA or some ratio of protein, which is defined by the ratio of mRNA. And we have this one mRNA which is most highly produced. Right? Okay, so this mRNA which is most highly produced has to be enough to accommodate all of the ribosomes. Basically, when will it stop being able to accommodate all of these ribosomes? When this number of ribosomes will be too high. When it will be too high? When the cell size grows. Because when we talk about optimal protein allocation, we don't care about size. Once we throw size into the business, then all of a sudden we also have the issue of how many ribosomes we actually have. Now, mRNA is not diluted just by division; it's basically rapidly degraded. So basically, the number of mRNA is not so much dependent on when we divide, but actually on how fast we produce. So we have this limit of how many mRNA we can produce, given this limitation. So we can accommodate this optimal allocation until we get to this critical size. Once we pass this, basically we lose, and then growth rate of the other things which we need to optimize for have been lost. So this might be another fundamental limit that one has to take into account, and this is sort of where I want to take you. And again, just to finish with acknowledgement, this work that I described, and again, the work of Eyal, Moshe, and Gilad. And thank you very much for listening and for having me.
Thank you, Naama. If it's okay with you, we'll just use whatever time that you have to work our way through the questions. If you have something else you have to get to, just let us know. But let's start. You've got a bunch of questions. So, Fred, would you like to unmute and start us?
Yes, I'm unmuted. Hello. I'm amazed that you get such high levels of expression. Are these levels now similar to ribosomal protein levels? And the other question is, if so, then is the cytoplasm getting more dense or crowded?
So when we compare to individual ribosomal proteins, then of course they are much higher than individual ribosomal protein. But overall, you have 100 ribosomal protein, or maybe 150. So we get, I think, to about equivalent amount. Now we look to see if there are granules forming. I mean, we looked at them by microscopy. We didn't see an indication of sort of darker spots or weaker spots. It looked, like I showed you in this figure, it looked good. Now we definitely have some problem because we see all this HSF1 module goes up, which has to deal with this, which has some problem with folding, etc. But we don't see any obvious effect on the morphology of or this density inside this.
So, Jan, you want to add something to that? Thank you, Fred.
Yeah, I thought Fred, that's an interesting question that Fred had. I guess I had kind of a related question where it's like, if you're now producing 2%, like let's say one copy of that gene you put in there, you're producing sort of an extra 2% of these useless proteins. Like, why don't we get more of a decrease in the growth rate? Like, because it seemed like you were getting, I don't know, maybe a % decrease in the growth rate, but you're adding like 2% of this.
Exactly. So there are two effects here, which I tried to allude to. The first effect is that the cells become larger. So there is some compensation here. The second effect, and because this is highly expressed gene, then it's increasing less than the others. So we get some minor compensation. But what we think is a more important compensation is the fact that the cells start using this extra ribosomes which we have. This is, I can go back to this figure. Quickly here, this one. So what you see here is basically, you see two lines, right? Just look at those 3 points. What those 3 points are actually showing you the relation between the fraction of ribosomal protein in the cell and the growth rate for different conditions. This is rich media, limited phosphate, limited nitrogen. Okay. Now, each of these 5 points is different level of burden. So this is high burden, this point, for example. So this is high burden in SC. And what you see is that this line of high burden is actually shifted down. Namely, it intersects here in different place compared to this one. So in our interpretation, there's always some fraction of ribosomes which are not being used, not because just the way that the cell is actually being engineered. And this fraction is lower when you put the burden, and this gives you compensation. This is why your growth rate goes down less than what you would expect with 2%.
Okay, so if I understand your answer, it's that you have kind of excess ribosomes to some extent. Yeah. And so by the addition of, like, let's say 2% more mRNA, it doesn't necessarily decrease the expression of the other proteins proportionally. Okay, thank you.
Now we have another indication that this is actually the case, because the prediction would be that this access of ribosome here is actually what you use when you go out of stationary phase. This is basically when you go to growth rate, you go to zero growth. Now you have to stay, you put back nutrients, you have to go out. For this, you need to start off, and you need some ribosome to start off everything. So the prediction was that the starting off depends on how much ribosome you have, basically you reserve. And those will take a longer time to start off. And they actually do, and it's proportional to this, basically, to this point. Okay, so different burden get to different point, and if you see that this starting time, how long it takes you to restart, when you go out of this zero growth mode, it's really proportional to this reserve. So this is basically, we think, support this interpretation.
Thank you.
I guess one quick question, just for a bit of context. How much bigger do the cells get, roughly? Like just say in glucose, when you really burden them?
Well, I don't have it just number. I think just look at it. This is cell size. It's 1.4, 50% larger. This is ST. It's 80% larger, and this is 40% larger.
Oh, cell size. Okay.
Yeah. This is for this given burden. It's proportional to how much burden you put in.
Right. Okay, so we'll keep going. Wei, would you like to unmute and ask your question?
Yes, thank you. My question is, presumably there are diploid cell versus haploid of the yeast case. Have you tried in different type of cells to see the growth burden has difference? Thank you.
No, we actually did it only in haploid cells. It's a good point. It's easier to work with haploid cells because, but we haven't done it in diploid cells. Diploid cells are not exactly 2-fold larger, they're about 80% larger to start with. But we haven't done this those games in diploid cells.
Thank you.
Gabriel, you're next.
Hi, very interesting. I have a question about the contribution of the ribosomal genes to your maximal transcription rate, because when we had a very similar observation when we grew cells very large and measured how much RNA is in there. We saw that most genes could be upregulated further, except for the very highly expressed genes. And so we had a similar thought. But then when I excluded the ribosomal genes from this analysis, I could not detect this anymore. And so I was wondering whether, I mean, you showed some data that the ribosomal genes are not affected, but did you try to do your analysis excluding the ribosomal genes?
Yes,
and would you?
Yes, the ribosome genes are exactly on the curve. They don't behave any differently in our case. It's not due to ribosome genes. You mean the genes coding for the ribosomal protein, right?
Yes, exactly. If you take those out and look at highly,
yes,
other highly expressed genes, you also see it.
Yes, of course, I don't have this figure here, but yes, we did it, of course. We looked at it. It's not coming from them.
It's very interesting.
So the next question we have is from Benjamin.
Hi, Naama.
Hi.
If I understood correctly, basically what you're saying is that growth is then at some point limited by DNA, the DNA of the most highly expressed genes relative to cell size. So would you expect that if you have mutants that reduce cell size, that these would rescue the growth burden, and the other way around, if you blow cells up, would they be more sensitive to protein burden?
So they will be more or less sensitive to protein burden.
If what is right?
Yes. So the question is, you see, I'm talking here, when I talk about this interplay, I talk about evolutionary interplay. What the evolution can do when it wants to optimize together the growth rate and the cell size, and to basically maximize cell size given some optimization of protein allocation. Now, I didn't claim that this is where the fitness of the burden is coming from. Actually, I don't think so. The problem of the burden cells, the fact that they are reduced fitness, doesn't come from this. This is sort of, we see it because we look specifically at the expression, and there is some limited from optimality, but actually the growing to become larger, we think it's part of the compensation mechanism in a way. So I don't think that this will actually change the fitness of the burden. Maybe it will make it worse, actually, because it will reduce some compensation.
Mm.
You see the last slide that I showed was motivated by our observation. This hypothesis that you have this basically maximal size given some optimality condition. It doesn't talk directly to the burden. It just comes from the observation that the burden cannot basically optimize the gene expression. We didn't say that all the fitness costs come from this. I don't think so.
Yeah, I should probably read the paper. I don't know. I mean, this question I had before you had your last slide, and I thought, which yeah. Okay, anyway,
I think that the
I'll
main
think about it more.
Okay, the main problem with the burden is actually a problem of protein allocation, and there is some compensation coming from being larger in size. But being larger in size comes with a cost that you need to also, or with a built in that you also need to increase your gene expression, and then you see this limitation that you cannot make mRNA as fast as possible. It's fast enough, so it's very high expression mRNA. This is sort of the logic that I try to paint.
Yeah. Need some time to process it. Thanks a lot.
Okay. Thank you.
Matthew, would you like to ask your question?
Sure. So given your model or this idea that some genes, the most kind of highly expressed genes, are capping out their expression in the kind of extreme burden conditions, I was wondering two things. One is, have you been able to measure the transcription rates globally in the burden condition and also on a kind of gene by gene basis? And secondly, have you compared the gene expression signatures in these burden conditions with the RNA polymerase mutants that are known to have kind of fast and slow elongating rates? And might you expect that those mutants, if you, for example, were to introduce the burden into them, if that would be possible, what effect that might have?
Right. So this is exactly the two experiments that that we need to do to actually get this more solidified. So we didn't measure transcription rate. We only measured mRNA levels. And the reason we didn't measure transcription rate is because we measure it in other contexts, and we don't think it's basically it's a noisy assay. So because of that, we didn't do it. But this is, of course, a problem with the argument that I'm saying here, or with this part of the conclusion. It could be that the degradation rate of those highly expressed mRNA is actually being affected, not the production rate, but actually the degradation rate. We didn't fully establish this. Not that we didn't try, but we didn't manage to set up a system that is reliable enough. For the polymerase mutant, we also tried. We actually have a precise prediction, but again, we couldn't get this reliably enough working in our hands. And this paper had to be published, and we had to continue. So this is sort of the 2 points that this work is really not complete. I think if we would have done those 2 points, our story would have been much stronger. We're still left with the question marks here. It could formally be that what we see is a change in degradation and not a change in transcription rate.
Thank you.
About the only thing I can say in my defense is that if you want the last point, the take-home message that about this limitation of that actually the cells work at this maximal capacity. This you basically can. It's indicated by our experiments, but you don't need it for experiments because there are other people that have measured this, and we discussed this is actually in the paper. And I think if you look at the number and also what people claim, this is, I think, something that we can trust. But actually, the cells go there. And if you count just how many proteins you have in the cell, we discussed this with this. So this doesn't depend on this point. But for our work, this is just not two missing experiments. I completely agree. I should have said it myself.
Thank you.
Thank you.
Okay, we have just a handful of questions left. Servidya, would you like to unmute?
Hi, sure. Sorry, I'm just trying to find my question. Is there any evidence of asymmetric partitioning of these useless proteins upon division? So like one daughter cell is getting more than the other systematically? And could you see an indirect signature of something like that in, let's say, the asynchronous population cell size distribution getting broader with protein burden?
Yeah, so this is a work that actually not this work, but it's related to what Felix was doing. He was looking at basically was quantifying. He was looking at the cell cycle in microscopy in those cells, budding is divide asymmetrically, and he was measuring the burden and looking at the cell size, and we didn't see any indication for asymmetric distribution. It looks like it paints a cytoplasm equally same way of both mothers and daughters. We didn't see any indication for non-symmetric distribution.
And the population cell size distribution is no broader than without this additional burden.
If it's not border, it's actually a little different. We can see some different. If it's border, it wasn't particularly border, maybe a little bit. We were hoping to see a border distribution, but it wasn't really the case.
Great, thank you.
Still have size control. Yeah, size control seemed to have been working. I guess we tried to prove that there is no size control, and we couldn't prove it. So it looks like they have a decent and kosher size control, even though we tried to prove the opposite.
Super, thank you.
That was a goal of the experiment, yeah.
Cells win, thank you.
Thank you.
We have a question from Marco.
Hi, thanks for the talk. I was wondering what is known about global regulation of transcription in the P and R sectors. There was a study in yeast. There was a study, I think by Aran Sigal a few years ago, showing that there was a common regulation from all the ribosomal promoters, but I don't know what sets this common regulation. And in bacteria, the common interpretation is that GPP does a lot of these first growth law stuff. So I was wondering what is known about how yeast implements the same kind of regulation.
I think in yeast, it's mostly not coming from something internal, but something coming from something external. There is a TOR pathway, for example, that regulates growth. It's mostly interpretation, at least this is our view, that it's mostly external regulation which sets the ribosome level, the mRNA production, not so much internal signals in the case of the ribosome. And for example, we can actually show you. I skipped this, but if you look at I showed you a very nice linear relationship between ribosomes and growth rate, right? Very beautiful. It completely, it also remains at the level of mRNA, so it's coming from mRNA. It completely fails when you look at mutants. So if you look at mutants, you take the whole stage data, lots of deletion, a broad range of growth rate, no correlation. You completely lose this curve.
But is there a specific, I don't know, category of mutations that cause this disruption, or it's like?
No, I think my view on this is that in budding yeast, the regulation of ribosomes comes from the outside. So basically, they sense the environment, they know how fast they should go in this environment, they use signals, they go on the up or down regulated ribosomes. So it's sort of a feed-forward type of interaction. You have the external signals, they define how much ribosome you're going to make, how much amino acids, they set all the protein allocation to be properly organized, and they can move on. Amino acids, actually, the production of amino acid biosynthesis genes, this is probably subject to autoregulation inside. Cells can sense how much, if they have depletion of amino acids. So this is sort of internal. But for the ribosome, my view or my I mean, we were thinking about it quite a bit, and my conclusion from the data is that it's basically a feed-forward type of mechanism.
Okay, thanks.
Thank you.
Okay, so I think Terry left. So we have just one question left from Kurt, and then we're done.
Hi, thanks for the talk. So I'm still a bit confused thinking about how growth rate plays into that. So if I understand your model correctly, if you have faster growth conditions where you have more ribosomes per genome,
yeah,
would the prediction be that optimal size should be smaller?
The prediction would be that optimized size would be smaller. I think it's not necessarily, because it depends, I think, on the allocation. So it should depend on what's the difference between the minimal mRNA and the maximal mRNA, right?
Right, but in my understanding is that in fast growth conditions you have more ribosomes, right? So you should probably also have more mRNA to keep the ribosome to mRNA ratio constant, which
yes,
would
so
mean that you need.
that would be, yeah, that would be my prediction. If the protein allocation remains the same, right? And now, well, wait, but no, it's nothing. I have this blackout for 1 second. You're saying, why is this has to do with the number of ribosomes? I'm only talking about the rate by which the maximally transcribed mRNA should be transcribed, right? It has nothing to do with
right.
which ribosomes I have. It's just how many, if you're saying that this making of ribosome is what's limiting, so you have an mRNA and the mRNA is packed with ribosomes, right?
Yes.
This is a proportionality. So I don't think it has to do with how much ribosome you have to do, but it has to do with the mRNA that needs to be, the protein that needs to be produced at maximal capacity, at maximal level. It could be ribosome, but it could also be, for example, something that codes for glycolysis, which actually—
Sure, but that would be produced by ribosomes, right? So as I was thinking about the mRNA to ribosome ratio for that maximally produced gene.
Right, but the level of the fraction of ribosome that need to be allocated to this particular mRNA, that would define the ratio of the ribosomes out of the total pool, and that would define the ratio. Yeah, it could be. Yeah, you're probably right. For a given protein allocation, I have to look at it more carefully. Maybe I missed this point. Could be, but it could be that you're right. I don't want to just throw an answer like this. We actually didn't think about it this way. Maybe we've missed something.
Because at least in the conditions that I know, like in general, the trend seems to be the other way, right? That if you have fast growth.
No, but this is something else, right? Because when you change between conditions, you also change the protein allocation.
Okay, yeah, so that would be your explanation. That
yeah, this is what I try to explain.
The size between media depends on the allocation rather than the.
It really depends on your protein allocation. It's exactly the point. The protein allocation differs between different conditions. And that's basically for a given protein allocation, this is how far you can go. Yes.
Okay, thanks.
Okay, thank you, Naama, for staying and for the extended question session. And thank you for everyone for asking so many questions. That's always what we want. Yeah, so that is everyone. Thanks everyone for coming here, and see everyone in 2 weeks' time. Thanks to the speakers too.
Thank you. Bye-bye.
Bye.